Hey friends, welcome back to my channel. In last video we have discussed the motion of lipid in cell membrane. So let's continue. Next is factor affecting membrane fluidity. So membrane fluidity is essential for membrane flexibility, protein movement and cell signaling. Next, factor affecting this membrane fluidity. First one is lipid composition, whether it is made up of saturated or unsaturated fatty acids. So this will decide the fluidity of the membrane. Second, temperature. So temperature is directly proportional to fluidity. High temperature, high fluidity; low temperature, low fluidity. Next is experimental evidence. So lipid movements observed using FRAP, fluorescence recovery after photobleaching. So it is a microscopic technique in which bleaching a small region with high intensity laser and then monitoring the recovery of the fluorescence as unbleached molecule diffuse back into that area. So by this we can observe the motion or movement of lipids. Next is transverse diffusion or flip-flop. So before that we have studied lateral diffusion. So in lateral diffusion, lipids move sideways in the same leaflet, but in transverse diffusion, movement of lipid molecules from one leaflet to another leaflet of the bilayer. So it is not spontaneous. This phenomena is very rare because it requires ATP. It is slow in process, takes hours to days. Then lipid polar head group must cross the hydrophobic core, which is energetically unfavorable. So this point is very important. So thermodynamic barriers prevent easy flip-flop. So it depends on fatty acid chain length and second on the degree of unsaturation. So we can say lateral diffusion is fast and flip-flop or transverse diffusion is slow and energy dependent. Now let's talk about the three enzymes Flippase, Floppase and Scramblase, which are involved in the movement of lipid or help in movement of lipid. So first one, Flippase, it is ATP dependent type, example P type ATPase. Direction of movement of lipid from outer to inner cytosolic leaflet. They are specific, example phosphatidylserine and phosphoethanolamine. Function, they maintain membrane asymmetry, moves lipid against gradient. Second one, Floppase, ATP dependent type, example ABC transporter. Movement of lipid from inner to outer leaflet, specificity is there. Function, also moves lipid against gradient. Third one, Scramblase, they are ATP independent, very important, bidirectional, non-specific, and their function is to equalize lipid distribution between leaflets, moves lipid along gradient. So here is the chart representation of all the three enzymes. Important point, asymmetry breakdown occurs during apoptosis. Apoptosis is cell killing or cell death. Scramblase activation, this means loss of asymmetry. Flippase inhibition, diseased condition. Lipid movement across bilayer is called transbilayer movement. This is important. Next is fluidity of lipid membrane. So membrane fluidity is ability of lipid molecules to move within the bilayer. We have studied this before. Essential for membrane protein function, transport processes, cell growth and signaling. Now let's talk about the effect of temperature. At low temperature, it is in gel state. Lipids arrange in gel-like ordered structure. Polar heads tightly packed. Fatty acid chain straight and regular. Membrane is thicker. Low fluidity, or we can say rigid membrane. At high temperature, we have observed the fluid state of the membrane. Lipids show fluid-like disordered arrangement. Polar heads loosely packed. Fatty acid tails are disordered or kinked, membrane is thinner, and high fluidity is observed. Next is phase transition. So change from gel to fluid state. This occurs over a narrow temperature range known as transition temperature or TM. So temperature is directly proportional to fluidity. High temperature, high fluidity; low temperature, low fluidity. Next is factors affecting membrane fluidity. First one, fatty acid chain length. Short chain, low fluidity; short chain, high fluidity and low van der Waals interaction. Long chains, low fluidity and high van der Waals interactions. Degree of unsaturation. Unsaturated fatty acids, they have double bonds, so they create kinks, prevent tight packaging, increase fluidity and reduce van der Waals interaction, and lowers the TM. Saturated fatty acids, they have straight chain, tight packaging, higher TM and low fluidity. Next is the role of cholesterol. So cholesterol is the key regulator of membrane fluidity. Cholesterol at low temperatures prevent tight packaging of fatty acids and increase the fluidity. Cholesterol at high temperatures restrict excessive movement and decreases fluidity. Structural effect of cholesterol. It interacts with saturated fatty acid chains, stabilizes extended straight chain arrangement, increases order and increases rigidity. Animal versus plant membranes. So animal membrane contain cholesterol, more regulated fluidity and often less fluid or more rigid. Plant membranes, they lack cholesterol, instead they have phytosterols and generally more fluid. So that's all for today. See you in the next lecture.